Hi friends, welcome to my YouTube channel Master Biology with Mariam Khan. Today we're going to discuss about a very interesting topic, that is antigens and organ transplant. You must heard about antigens and antibodies, but today we're going to study what are basically antigens, antibodies. So first we're going to talk about antigens. So all cells have antigens on their cell wall, cell membrane, and every person has a unique set of antigens on their cells, tissues, and organs. And in our blood, white blood cells known as lymphocytes, they actually produces antibodies in response to antigens. Therefore, they have to play a very important role in recognizing the self-antigens and foreign antigens. So, antigen are basically any substance that causes our immune system to produce antibodies. And antigens can be proteins, they can be polysaccharides, they can be conjugates of lipids with protein, like lipoprotein, or conjugates of lipids and polysaccharides, like glycolipids. So, antigen can be any foreign substance from the environment, like it can be chemicals, bacteria, viruses, or pollen, which induces our immune system to produce antibodies. And antigen may also be formed within the body, as with like bacterial toxins or tissue cells. So, first we're gonna differentiate between antigen and antibody, right? So we have discussed that antigen is any substance which induces or prompts our immune system to work against it, whereas antibodies are basically protein. They are produced by white blood cells known as lymphocytes. And they are made from polysaccharides, and they can be a protein, polysaccharides, or conjugates of lipids with protein. But antibodies are purely protein. So there is no other molecule attached to it. And antigens can be cells, like bacteria, virus, and these all pathogens, these are cells. So they have those antigens on their cell surface membrane. Whereas antibodies, they are not cells, they're just proteins, right? They're just the molecules, biological molecules. And antigen, basically, when antigen and antibody interact with each other, so antigen acts as the key and antibody is the lock. So the key and the lock joins together, and the white blood cells, lymphocytes, basically combines with the antigens and destroys them. So there are two main types, like we call antigens as self-antigens, which are autoantigens, like the antigens on the person's own cells, tissues, and organs. So lymphocytes will not produce against them antibodies. It will not destroy it. And then there are antigens which are foreign antigens, which we call as non-self antigens. So lymphocyte job is to recognize those non-self antigens and produce antibodies. Whereas antibodies are like five according to the protein constructs, and these are like IgA, IgD, IgE, IgG, and IgM. Okay, so here's this picture which shows antigens on the surface of pathogens. Pathogens are like disease-causing organisms like bacteria and virus. So you see the antigens on their cell surface membrane and cell wall. It can be cell membrane or cell wall, depends, right? So here we have a structure of antibodies. So antibodies are like Y-shaped structure you see here, and these are the binding sites where antigens bind, and they are a Y shaped, and they're composed of two chains. One is heavy chain, you can see these are the heavy chain, the blue one, and these are the lighter chains, right? And on the tip of the heavy and light chain here, antigens binds with the antibodies. So this is the binding site of the antigens. You can see here antigens bound with the antibodies on these sites. Oh, this picture shows a very important role of white blood cells against pathogens. You see this one, this is the white blood cells known as phagocyte. It has lobe-shaped nucleus and it is larger in size, and 65 to 75% of the white blood cells composed of phagocytes, right? And it basically, what does it do? It engulfs pathogens. So they just form a pseudopodia like structure around the pathogens and engulf it, and then later it inside the cell it produces digestive enzymes to destroy the pathogens. So that's how it destroys pathogens. The process is known as phagocytosis. So it basically phagocytosis means eating away. So basically it engulf pathogens and secrete digestive enzymes to destroy pathogens. But here are another white blood cells which are called as lymphocytes or B cell, right? So it produces antibodies which attack against antigens, and we have studied the structure of antibodies. They're like Y-shaped structure made up of two chains, and they have antigen binding sites as well. So what happens, these antibodies, they bind with the antigens, and they either clump them in one place, all these pathogens or antigens, so it will be easier for phagocyte to engulf them, or it either destroys the antigens. Now we have studied about corona a lot, and scientists are still studying about it, and there are vaccination being done, and vaccines being made to protect the individuals from corona. It's a novel disease caused by a virus, and it's a very obviously it's a very dangerous disease, because viral infections are always dangerous. Like it can be like you have to protect yourself from the vectors. Vectors are any organism which are carrying the pathogens. So you have to protect yourself from the vectors so that you don't get it, right? And once the virus gets into your body, then you become the vector, and then people have to protect themselves from you, and you also have to protect yourself from others. So antigens are structural parts of a virus. This is the structure of a coronavirus. It has spikes on their surface, right? So these spikes are basically used to adhere to the cell membrane of organisms, right? So during vaccination, and what happens in our body, white blood cells produces antibodies when somebody's get vaccinated. So the white blood cells produces antibodies. So when the person is exposed to the real pathogen, or when virus enters in their bodies, so antibodies are already there to fight against these antigens and these pathogens. So these antibodies, they will be blocked by, they will actually block the antigens present on the surface of the virus, so that virus cannot adhere to the cell membrane of other cells and do not infect other cells. So antibodies block these spikes so that they cannot adhere to the surface membrane of other cells. So that's how vaccination gives us protection against diseases. Transplanting an organ or tissue. So, like during transplantation from a donor organism, any cell, tissue, or organ is transplanted to the recipient organism. And our lymphocytes lymphocytes will recognize them as non-cells, and it will start producing antibodies. So it will result in organ rejection. So therefore during transplantation, what happens is that we're always looking for closely related family members, or with whom our blood group matches. so that we don't face organ rejection. And this process is called as the tissue typing. So a person's antigens, they are determined in their genes. So tissues from family members are a very good, they're a good match, right? So and if a person cannot have a good match in their family, so tissues and organs from people who are not related to the patient can be tested to see if they match those of the recipient. And if it happens, then organ chances of organ rejection will be reduced. And also during organ transplants, so there are some drugs given to the patients which are called as immunosuppressant drugs. So it actually stops lymphocytes, white blood cells, to produce antibodies against the antigens which are present on the transplanted organ, tissue, or cell. So tissue typing and immunosuppressant drugs basically reduces the chances of organ rejection. And this way, we can have a successful transplant of any organ, tissue, or cell.